I'm John Poole, and I'm here to introduce Myron Child, who I am very glad to have as a PhD student in my lab. Um, I was thrilled to recruit Myron. Um, he was already a very experienced researcher with a lot of really relevant background in Drosophila especially. And he has done a great job in my lab. He's overcome, an issue with his first project that had nothing to do with him, but um, came up nonetheless. And so he has been rebuilding that data set and, making good progress on two other major projects. One of which I think you'll especially hear about today. And this is somewhat in its early stages, but I think has some really exciting potential. So with that, go ahead, Myron.
All right. Thank you very much. Okay, yeah, so I'm gonna talk about a somewhat newer project, if anyone has seen me present before, that I've been working on just over the last year, where we're looking at fruit fly pheromones and how the forces of adaptation and sexual selection might be potentially working in different directions and how that affects wild-derived fruit fly populations. So in general, the kind of questions that I'm interested in when it comes to evolution are how new species form. And so very simply, you have these forces of drift, adaptation, and selection that work to create genetic variation in a population that eventually is enough that you have distinct populations that are no longer able to reproduce due to some kind of reproductive isolation between them. But what I think is particularly interesting is what happens at these early stages and how do you get this initial separation that's enough to keep them from interbreeding and just fully changing their genes. And a particular interesting way that you can have interactions here is with adaptation and sexual selection being opposing or similar forces in this aspect. So I'm going to talk a little bit about how these might be interacting in a specific example in Drosophila. So to understand this story, I have to explain cuticular hydrocarbons. So cuticular hydrocarbons are these sort of fatty lipid molecules that are produced by the fly, and they live on the cuticle, hence why they're called cuticular. So I'll shorten these often to CHC. And they have two main functions in Drosophila. The first, as I've alluded, is that they play a role as pheromones. So they can be both sensed, like smelled and tasted by other flies, and these are important mating and kind of sexual signaling cues. So males and females have different sort of profiles of what cuticular hydrocarbons are present on them and what ratios, and a lot of times the difference between them is just sort of simple length differences. But they're very important for both sex interactions and different species interactions and identification. But then the other important role that they play is, as I've said, these are sort of fatty lipid molecules that live on the cuticle, which means they affect the transpiration of water in and out of the cuticle. So they're really important for how flies are retaining water. Turns out fruit flies are actually really prone to dying under dry conditions. So here you can see if you actually remove these cuticular hydrocarbons from the fruit fly by sort of ablating the cells that produce them, their ability to survive in dry conditions is very poor. They all die in a matter of hours. But if you just add back these CHCs, either through artificially perfuming them, just kind of spraying them on top, or even through just interacting with other flies enough that the CHCs can rub off on them, it's sufficient to really drastically increase their survival again. So we know it's important for both of these roles, but how exactly these things might be interacting is there's still a lot of questions we want to know. We know a fair bit more on what actually happens with these cuticular hydrocarbons on the female side of things, and we actually have specific genes that we think are responsible for some of the main differences between females that we observe in wild populations between ratios of cuticular hydrocarbons. But we know comparatively less about the males. What we do know is that in males, there are two main components that make up their sort of profile of what kind of cuticular hydrocarbons are present. And the most abundant is this first one, which is called 7-trichosine, which I will often just call 7T. And this is the main, most abundant CHC present in males um, uh, in most cases. And it's been shown experimentally, people looked at this because it's so abundant that it's responsible for increasing female receptivity. So having it makes you more likely to mate with a female. And it also works to deter male-male courtship, which will be not fruitful. But the other main prominent component is the 7p here, which is 7-pentacosine. The difference between them is one is just basically a slightly longer version of them, hence the name here. And in certain populations, particularly those from West Africa and the Caribbean, this actually is the more predominant CHC in their sort of total profile. This pheromone has also been shown to elicit female interactions and make males more likely to mate with females. But for whatever reason, it's also been shown to increase male-male interactions. So males, rather than being discouraged by 7T, are actually encouraged to mate by 7P. Don't really know why that is the case. But people seeing this have tried to examine what could be going on here. And so there's some evidence that the level of 7P you have, so the one that's more predominant in West Africa, might be important for desiccation resistance, though the data on that is a little sparse. There is comparatively better evidence that females are actually pretty good at discriminating based off of 7T and 7P ratios, with a general trend being that females from a population that have more flies with 7P tend to prefer males that have that similar ratio. So we know this going into it, but this data that I was showing before is on two specific kind of strains of fly. The first one is a cosmopolitan one that's used in a lot of labs called Cantonus. But the other one is one that is specifically from West Africa. It's called Thai, confusingly, but it's actually from the Ivory Coast. It's just named after the place it was collected. So this is a West Africa line. And so you can see these big differences in their ratio, and you can quantify those as sort of how much 7T to 7P do they have. And so a positive ratio is going to be more 7T, which is sort of the more cosmopolitan. And then a negative ratio is more Western Africa-like, so more 7P. But we have more than just these two lines that we know about their particular hydrocarbon information for. And so if you actually look at across lines from Africa, you'll see there's a good amount of variation, but that trend still holds that Western Africa, so these Nigerian Gabon lines here, have more of the 7P, that West Africa phenotype. And then other flies from different countries in Africa still have more 7T, similar to what you see in like European populations as well. And so this work was collected by a collaborator, Henry Chung, who's at Michigan State, who was helping us look at these lines we had and helped us realize that there's some cool potential to look at here. So given that we see this variation, we know it's potentially important in all these different ways. We wanted to ask, what is the evolutionary genetic basis of this polymorphism that we see in wild populations? And so the first thing that we did is John Poole here did a population genetic scan using some of these wild populations where we see genetic differences. And so this is a population genetic scan using FST, which just sort of compares the genetic differentiation between two populations. In this case, we're doing a West Africa population and populations from Zambia. So it was one of the other African populations that doesn't have the same profile as Western Africa. And so if you just look for outliers on these, you find a few that are important. You find a few that are significant, particularly on the second chromosome here. But the reason that I've highlighted these specifically is that if you look more closely, and actually follow up with a little more detailed scan, population genetic scan. So this one is a population branch statistic, which tells you a similar kind of thing. It's kind of a rescaled FST based value, but allows you to kind of compare a focal population to two outgroups, and it helps when you're looking at a lot of different loci like you would be across the genome. And it also helps kind of tell you which one is more, you can compare one to two outgroups, which helps you tell which population you think might be experiencing local adaptation, thus leading to different genetic or leading to genetic variation. And so if you zoom in on these regions, two particular genes stand out, not just because of their significance in the statistic, but because they have predicted functions that seem particularly relevant for cuticula hydrocarbons. So this one on the left chromosome is predicted to be a reductase gene from what we know from other fruit fly genetic information. And the one on the right side is an elongase gene. And they're predicted to work through this sort of this fatty acyl coenzyme A reductase sort of pathway. But why that's relevant is that if you'll remember, a lot of the differences between pheromones is just sort of a length difference. And the actual difference between 7T and 7P is that one 7P just has like 25 more carbons in this lipid molecule. So it's the fact that we have two genes relating to how long your lipids are being produced seems particularly relevant here. Again, Henry followed up this information with kind of some functional work where you can stain just the cells where cuticular hydrocarbons are produced using a specific kind of antisense probe to these genes. So these are the ones that were labeled on that plot before. And you'll see one that they are present specifically in these cells where cuticular hydrocarbons are produced, and they seem to be present in variable amounts depending on whether you're looking at the more cosmopolitan, that's 7T heavy, or the West Africa 7P heavy. And you can also do, he did this kind of quantitative PCR where you can look at the relative expression of these two genes in the difference between these two lines. And so you see, in general, this Thai Western Africa line has more expression of both of these genes, but the important takeaway is that they're significantly different from each other. We also have some RNA interference evidence that suggests knocking down these genes actually does change the levels of expression. Change the level of the cuticular hydrocarbons present in the profile. But I won't go on to that here because this was done by our collaborator here. But so given this, we know that there's seems to be good, strong evidence for these genes being important in the phenotype that we're trying to look at. So we want to try and experimentally confirm their effect. So the goal of my project is to just make two genetic mutants using these specific genes. And then what we want to do is actually try them in the wild-derived lines where we know there are phenotypic differences in the CHC profile between them in a more relevant kind of evolutionary genetic context. And then once we have these mutants, we can test their effects, depending on if we're looking at a high 7T or 7P ratio fly. So we can look at the CHC profile, and then we can also test those particular phenotypes that we cared about. So how does it affect the mating behavior and success? And how does it affect their ability to survive desiccating environments? So just a quick rundown of how we sort of propose to do this in this context. So we'll use CRISPR on these lines, which is a little more difficult because they're not these kind of common lab strains, but you can definitely do it. You make a null mutant in one of the genes that you care about. So we had these two that we're looking at. And you just do that by inserting a fluorescent reporter in place of the gene. So the gene is no longer there, but we can track whether or not we had a mutant. And then we will repeat this for both a high 7T and a high 7P line, so the Western and kind of non-Western phenotypes. And then we actually want to test this as using something called a reciprocal hemizygosity test. So in this case, you have a mutant in, say, your high 7T ratio line. You would cross it to the other line that has more 7P. And then this F1 that you get from that cross is hemizygous, and where the only functional copy of the gene 1 that it has is from this negative ratio 7P line. And then you repeat the same thing, but for the other population, so in this F1 hemizygote, you have just the functional copy from the positive ratio line. And then when you go to compare these two F1s to each other in a reciprocal sense, because you're sort of doing it in opposite directions, the only difference between them is which functional copy of the gene that they have. So any differences should be particularly just caused by these different alleles. And then, of course, this is for the gene 1. You can repeat this then for gene 2. We'll use a different colored fluorescent reporter to keep track of those different, but the exact same reasoning applies there for the other gene. And then also, given that we suspect there's probably some sort of epistasis between these two genes because they're working on a similar type of phenotype in different directions, you can also just do another kind of hemizygote, but for both genes at the same time. So we can examine double mutants and see what the effect there is. So that is the kind of experimental plan here. In order to do this, the first thing we have to do is pick lines that we want to use. So we need a positive and a negative ratio set of lines to use. So this is the data that I showed for these populations before. But this is sort of some average data from across these populations, and we actually have multiple individual lines from each of these populations. So the thing that we care about in picking wild lines to use is just basically whichever ones are the most differential between, whichever has the biggest differences that we can tell between. So it turns out the ones that we've tested, we have a specific line from France that is really, really skewed towards having 7T. And then the most skewed towards 7P is actually still this tie line from Ivory Coast that we had looked at before. So these are the lines that we are using for these projects. And before we do sort of the actual transgenic experiments, we want to test what actual phenotypes we see in these parental populations to confirm that there's actually something to look at there. So the first thing we did is we took these lines and we looked at the mate choice. So for this experiment, we simply take a female, give her a choice between a male from the 7T population and the 7P population, and see which male she mates with first. And so if you do this experiment, the general trend that you'll see is that the West Africa females, so these Thai females, really strongly prefer males that are Thai. So at about a 70% preference here. And then the France don't seem to have a really statistically significant difference. There is a slight preference for Thai here, but not that you can say statistically different from random. And the preference between France and Thai females is significantly greater in the Thai females. So we know there's a difference in preference here, which is good. Otherwise, it's hard to test a loss of function in these alleles. Then we wanted to look at the desiccation resistance. So to test desiccation resistance, we're just simply asking how long do flies survive if you give them a really dry environment. So we tested 1,600 flies for this set of data I'm going to show you. They're just put in a chamber with less than 5% humidity that we kind of experimentally confirm. And then because these flies are from different locations, not only does the humidity vary in those environments, so does the temperature. So temperature likely is a pretty important factor in how you are able to survive in your environment. So we also tested multiple temperatures for these. So first I'll show you the male data from this for our two lines that we've tested. So the color here is going to be how hot of the temperature we used. And then you'll have the dashed line, which is our Thai line, our West Africa, and then the solid, which is our France line. So the first thing that you'll notice is that indeed our Thai lines, our West Africa, are surviving much longer than the France line. And this is regardless of the temperature, with the exception that the hotter you get, the kind of less difference we see between the two lines. So those, we see a significant difference there. In the females, we also see a significant difference that again is temperature dependent. We can still kind of distinguish the lines at really high temperatures, but the difference is much smaller. And Thai is consistently surviving longer than the France females are in this case. But particularly if we're caring about particular hydrocarbons and we know that they're different between male and female, it's useful to look at them on the same plot together. So the other thing that you'll notice is females survive significantly longer than males. This is partially due to the fact that females are just larger, but there could be other important sex-related factors that we're not 100% certain on what they are. And the female differences are actually greater than the differences you see between lines. I've also just sort of said that they're generally different from each other, but you can statistically confirm these by kind of doing median survival estimates, and you can get p-values that tell you they are significantly different from each other. So we know there's a difference here. The other thing you'll notice is the fact that females still have a difference between the lines suggests that the difference between 7P and 7T that we're seeing is only present in the males. So that can't be accounting for what is happening for the difference in survival between the females. So it could be that females have a different set of cuticular hydrocarbon differences that are making them differentially adaptable, or there could be some other set of functions, phenotypic things that are making them better adapted in the Thai line, the West Africa line, or probably some combination of both. But if the cuticular hydrocarbons are responsible in the male, we should see a reduction in our mutants between this difference in the males and not the females. So we know what we want to compare. One thing I want to point out, if you look at this data, is data that looks at survival over 25 hours. The way that you do that is by sitting and watching the flies for over 25 hours, which takes a lot of time. So when you're doing experiments like this, you really need to be careful and precise about the kind of data and experiments that you're taking. So this is, I think, a good time to talk about rigor and reproducibility in your experimental design, and making sure that you're keeping track of important variables, testing them, and having appropriate controls, and then also just documenting everything that you're doing. So going into this, some things, if you look at through the literature, people do pretty wild and very different types of desiccation assays, different shapes, different amounts of flies, different ages, different types of desiccant. But if you look at conclusions from all of those papers and kind of aggregate them, there are certain things that you expect to be important in terms of their desiccation survival that we knew beforehand. So we know that species matters. Your sex and your weight, which are related because females are larger than males, affects how fast you die. The age of the flies certainly affects them. If they're too young, the cuticle hasn't fully developed. If they're too old, they get smaller and they're like a little bit thinner. The time of day that you do your assay matters, as well as just the density of larvae during the development of the fly. So actually before you even do the assay can affect their size, particularly. The temperature during the development of the flies before the assay affects what type of cuticula hydrocarbons are produced by the fly. So that's something you need to keep constant. And then also, as I alluded to before, whether or not the flies have mated can affect which cuticula hydrocarbons are present because they can exchange them between flies. So we knew this going beforehand, but in doing this testing and looking at results and being confused and redoing and testing a bunch of different variables, we actually found that there's several other things that weren't necessarily known beforehand that are really strongly affect your desiccation resistance. So fly line, we had some information beforehand, but I think this is a pretty strong clue of that, even just looking at 2 lines. The hydration level of the food, you would think might be common sense, but we didn't think of it originally, and other papers don't really report about it. So we specifically make food at a certain hydration and a certain amount of food ratio, and specifically at a certain day of age for the food. The amount of desiccant and the quality of the desiccant that you use in the assay can really significantly affect the actual humidity of the chamber that you're testing. A lot of papers don't report on this at all. They just say that they did some kind of desiccant amount and they said that it's good and that it's probably less than 20% humidity. So they don't actually report very clearly. As well as the density of the flies in the assay, so how many are in the chamber, and the size of the chamber can affect because they're interacting in different ways, and also affects the actual humidity of the chamber in that way as well. So all important things that took a lot of work to discover were relevant. And in trying to compare to other papers makes it really hard to actually compare your results to past ones. So the particular makes it hard when you find a paper where you're looking like this and they look at desiccation resistance across populations, and they have these nice stock numbers that you can compare for all these lines. Then you get to their melanogaster strain, and this was just a gift from the Caro lab, which is, I'm sure, a very lovely lab to get flies from, but I don't know what those are. Hopefully they remember what they were if we wanted to try and check them, but not the best way to be consistent in your data. The other thing in rigor and reproducibility is, so on the mating behavior I showed you. So the way that we did this assay is you have males from each population, and you can just coat them in this sort of fluorescent pigment, which is actually pretty easy to see and remains on them for quite a long time. And there's been evidence showing that this is not affecting their behavior significantly, whether you do or don't do that. So you take a male from each population, cover them differently, put them in a vial with a female. You see which male the female chooses to mate with. And this is what that data looks like. So you can actually see the pigment on the flies here. So this is a green fly and a pink fly. And so you can just see which male the female chooses to mate with by the end. But what I didn't show originally is that this was not the first run of the data. This was our second trial. So in trying to make these experiments a little higher throughput and a little bit easier to do, we thought, why not just put the 2 males in the trial beforehand instead of having to put them all in individually one at a time? Because the way you have to do that is you have to aspirate them with a sort of mouth aspirator, and it's really easy to lose a fly, and it takes a long time, and you have a limited window to test them because the time of day matters and all of that. So we put the 2 males in a vial beforehand, and then on the day of our assay, we just take a female, put her in the vial, and see which one she chooses to mate with. However, when we did this originally, this is the version of this data that we got. So the strong preference that we see is pretty much totally gone, at least statistically. There's still some preference that Thai has for Thai females, but that difference is pretty much gone. So there's a couple reasons why this could be. The version of the assay that we ended up going with, if you spend some time thinking about it, does make more sense sort of ecologically and for the variables we want to test, because sort of being forced in these vials for a long time is sort of an unnatural condition the two flies are forced to interact with each other a disproportionate amount. They're in there for a few days before we did the assay so that you could ignore the effects of anesthesia, which also affects their behavior. Anyway, the final version we went with is a better version of the assay, but something that is pretty easy to overlook if you didn't know that it would be causing a problem. Why exactly it's happening, it's hard to say. One interesting point is I'll note, like I suggested, the 7p has been shown to increase male-male interactions. And if you actually look at these assays while they're going, you very frequently see, so in this case, the pink one is a 7t male. And instead of trying to court the female, he is courting the other male. So he's actually actively trying to mount this male who's already mounted the female. And so you see a lot of examples of this. So it could be that the males from our 7t line are just spending so much time chasing or interacting with the males from the other line that they're altering their CHC profile ratio, or perhaps they're just getting it all over the vial that we're testing. Hard to say exactly. But the takeaway from here is that, so for behavioral factors, the thing that really is important is literally everything. Behavior is wonderful. I love it. Yes. So again, just documenting all of the things that you're doing and keeping track of your important variables is a necessity. So this is all data on the sort of unaltered lines that we're going to be making mutants in. So once we have those lines, which we've been sort of waiting a month and a half at this point for some reagents, shout out Workday, we can basically just repeat these experiments. So we compare the CHC profile of the mutants as I outlined before. There's also a chance that there's some sort of dosage effects, so you can see if that is relevant by looking at the CHC profiles, considering these are just going to be hemizygotes. And then we repeat the test for behavioral and for resistance and see if they're different in our mutants versus what we saw versus our kind of expectations as well. And then additionally, in doing these desiccation experiments, we think there's some really cool, interesting results that we can pull from that. But Thai and France are not the only lines that we have in the lab. We actually have a ton from different places that have been collected from the wild in Africa. And so I have a kind of a side follow-up project that I'm doing with an undergrad where we're just trying to look at desiccation resistance across all of these different populations and see what kind of things we can quantify, see if we can, if there's, particular genetic variants that might be related to this. If the more West Africa you matters, if there's a lot of variation within West Africa, how it compares to other lines and things like that. And so this also includes some new lines that really haven't been characterized before that I actually collected a couple years ago from the Gambia. So we have some new lines that are kind of the most West Africa you can get. So that'd be really cool to see those in comparison. So with that, I just want to acknowledge everyone in the pool lab for their help. Colin, his undergrad, is helping me with these desiccation experiments and helped me a lot in figuring out the variables that mattered. Henry Chung, who's our collaborator for all the CHC things, my thesis committee, and then also the crew that I had helping me collect flies in Gambia. So with that, I can take any questions. Yeah.
So when you were looking at genetic variation in like the two genes, the longase and reductase genes you identified, do you see variants that would rise to the level of a null mutant? Have you looked at potentially doing a little bit more of a change of function or something closer to what you see in natural genetic variation instead of just
yeah, so I mean, we like that information was just gathered by looking at genetic variation given those statistics between populations. We can actually look at sequences of specific lines and see what base pairs are different in those genes. There are definitely variants between France and Thai. I can't remember exactly off the top of my head. In one of the genes, all of the variants we see are sort of in the 3' UTR region of the gene. Another one, we see several variants actually in the coding region. There's also some kind of interesting patterns in terms of how many SNPs you have inside and outside the gene. So you might have a significant number of SNPs in the gene. I think it was the reductase gene. And then immediately outside, you see absolutely zero SNPs at all for quite a while. So there's definitely some interesting signals. Obviously not like, whole loss of function of the gene that we expect. So, in theory, our thing is this genetic mutant is a stronger phenotype in specific reference to those genes, but how it overall affects the 7T, 7P phenotype, we don't know yet. Yeah. I mean, it'd be cool to, so there have been papers that look at desiccation across a bunch of different species. In my opinion, they didn't actually do their assays very well. They looked at just the last time a fly was alive for those species, which is, I think, really prone to bias in a lot of weird ways. There's a lot of variation between them. Drosophila is particularly interesting because it is everywhere. So we have all these unique populations in Africa and a bunch of places in Europe and the Americas, and most of the genetic differentiation actually is within Africa. So there could be a lot of interesting things in other species. Genetics get a little bit harder with that. But if you found, I think a better approach would be if you found really interesting things, you might then try and look at other species that are really different in those genes or other types of things, and then try and do similar experiments in that. So I think this is the easiest way to check the effect of those genes, and you could follow it up for like more confirmatory evidence. But yeah, it varies a lot between species. So there's also more variables that affect that in species, like huge size differences between other species that are not going to be captured just by those genes that are known to affect desiccation. So maybe not directly as relevant of a test, but yeah. Well,
I have a two-parter, right? Is it fair to think about the Thai France comparison of meat print as sort of a loss of this preference for strain specific, like
yeah.
speculatively, and then like what might kind of contribute to the factor of that?
Yeah, the directionality of that is hard to say exactly. So ancestrally, flies are from Southern Africa. So they're actually ancestrally from Zambia, which is one of the lines that we tested, which is more 7T. They've then expanded up into other parts of Africa and then out of Africa. So everything sort of above the Sahara that's expanded into like Europe and Asia and all that are pretty genetically similar to each other, especially compared to the African lines. So it's hard to say exactly what the directionality is. I guess the sort of implicit assumption is that there were different conditions in West Africa, so they've changed from the ancestral, but that wasn't favorable going other places. That's assuming that desiccation is an important factor for the CHC profile, which may end up not being, and it might be more a behavioral thing. So it's hard to say exactly what the directionality of that could be until we know more about it. Yeah. But the idea is that it's known that different lengths of hydrocarbons can affect your ability to survive desiccation, that like resist water transpiration more. And then they also can really, really quickly vary given like sexual selection, mate preference variables. So there's other populations where there's other Southern African populations that have a really strong preference for their own set of males, and they like will almost never mate with the kind of cosmopolitan males, but they don't have this same ratio. So that there's multiple things. Yeah. Yeah.
Yeah, I was curious. The females that you put in the vials, were they virgins or were they, or did they mate before? And also like in the same way, do you think their mating preference would be affected by which males they had mated with before? That is either Thai or the Western one.
Yes. So the question is just on the mating assay. The females are always virgin because it has a really strong effect on their behavior if they've mated before and who they've mated with. So yes, it very important, matters for a lot of things. So yes, they are all specifically virgin. So are the males, because whether or not the males have mated can also affect things, and how the males are housed can affect their behavior. So yes, you have to be very consistent with all those things.
Do you plan on doing certain like this kind of assays where you would like let the female mate, like the Thai female mate with the trans one, and then try to see what they prefer and vice versa?
I mean, it would be interesting. It's probably like just experimentally feasibly more work than I'd be able to do. Like it would be really interesting though. Yeah, to try mating and see if that affects their preference. I'm sure it would. I don't necessarily know what direction, but yeah. Okay, quick more question. I don't want to take up Manny's time.
But the females are why? What about the 7T they're responding to? Like, is it they learned that's the CLC of choice because that's what their friends had growing up? Or?
Yeah, it's hard to say. I mean, people know that females have really strong preferences for certain CHCs that vary between populations. But why exactly that is, it's hard to say. I mean, certainly there's like very likely some sort of sexual selection role where they're preferring that the males that they're interacting with. It could get more complicated if you had kind of, if you have like allopatric and sympatric species, you can test that a little bit clearer. But the fact that these are, you know, just sort of populations that are somewhat isolated from each other makes it hard to test those kind of, you know, divergent forces that you might be having. But why they why these ones like 7p, I have no idea. You know, they're very similar. And it's not that they don't have the other one, they just have more of the 7p than the 7t. And it might be the case that the France do actually have a slight preference for that as well. It's just not technically significant. So maybe there's something inherent about that, but is problematic in the environment that the France flies are in. I don't necessarily know. Yeah. Yeah. Okay. I don't want to take too much time. Thank you.